All right, we'd like to start this morning off with a bang. We have Steve Williams, Chief Medical Officer of SomaLogic, and he's going to talk to us about Aphrodite and Medusa.
Thank you. Good morning, everybody. Wow, you actually answered. I'm going to talk today about a bit about beauty, which we're representing here by Aphrodite, and unfortunately rather a lot about monsters, which are represented on the right by the Gorgon. The point here is that these things are complicated. And as humans, what we try and do is make them simple. We heard that was one of the definitions of intelligence yesterday. But sometimes we get kind of carried away, and I'm representing here the idea that maybe you can capture bits of beauty and bits of the monster by some surrogate, some biomarker of the biology or of the disease. And the trouble with this is we kind of get carried away. We make the mistake sometimes that a little piece of biology is really representing the whole thing, or that a biomarker we like is actually truly causal, and what we need to do is to get rid of it. When I was doing my Ph.D., my supervisor, who's now Sir John Tooke and Dean of UCL London, he had a lovely picture of a motorway pile-up. And he said to the audience that every time you look down on a motorway pile-up, there are these vehicles with blue flashing lights on them. All we need to do to prevent pile-ups is to make sure that no vehicles with blue flashing lights ever travel on highways. And that's the causality problem. So we get disappointed. We get disappointed when our favorite marker, when, with its blood pressure or HDL or whatever, it fails in some way. And some of that disappointment is silly because we actually should never have believed that we were capturing the entire beauty or the entire monstrosity with just one marker. And Peter Gans is going to come next, and he's going to show you how one marker can capture an important piece of the biology. But most of my talk, I'm going to assert that if you really want to capture the true horror of the monster, you have to build together a coherent pattern of the biology or of the monster. Now, an exception maybe to this has been in HIV. Counting viruses in HIV has been one of the most successful biomarkers of all time. And people have argued that, well, the reason it's so great is because it's causal. And undoubtedly it is causal. Without the viruses, we wouldn't have HIV. But another reason it's successful is that we've reduced this down to one dimension. If you count viruses and you have a drug that kills viruses, we've effectively made the number of dimensions of success one. So I put it to you that another reason why HIV is counting viruses is successful is because we don't expect it to do anything else. We don't expect virus number to predict the complications of drugs or cardiovascular outcomes in HIV. We restrict our expectations because it's causal. Which brings me to Medusa. Isn't she lovely? Does anybody know how many serpents there are on Medusa's head? No one knows, because actually nobody does know. And that's the point. That is our job. We've got to find out for each Gorgon that there is in each disease, we have got to find out how many serpents there actually are. Same with biological beauty. How many dimensions does this have? So the point here is that instead of just measuring one thing at a time, let's build up a model. Let's arrange our Lego bricks here. This may not be quite as beautiful as the statue, but it's an awful lot closer than a single block. And so what I'm going to talk to you about today is how you can build up a representation here of biological truth. It's not quite the whole truth, but it's an awful lot closer than we are if we do a one-at-a-time list of favorites. But there's a challenge here, and I'm going to talk to you mostly about proteins here. The challenge is that if we want to build up this complicated picture, we've got to have a complicated technology that measures many things. And if you're measuring many proteins, you actually have to cope with a billion-fold range in concentrations. And this has been the problem with proteomics, that you've had to choose. Either you can measure lots of proteins over a narrow range, or if you want to measure the whole range, you have to measure fewer of them. And the technology that Larry has spent the last 20 years working on actually overcomes these issues. And the way that Larry and the team, before I joined SomaLogic, overcame this issue was to create a new class of reagents. And you're looking at a picture of one. The snake thing that you see is a DNA, a length of DNA about 50 bases long. And then the pink things that you see sticking out there in space, and it's kind of an impressionistic diagram because they don't look as if they're connected to anything, which to the purist is a problem. But to an impressionist, the idea is that these pink side chains that stick out of the DNA that Larry and the team at SomaLogic added to DNA, they improve the binding. And until we had done some crystal structures, we couldn't prove exactly why that binding got much better. But in the picture you see that the pink side chains are actually responsible for a lot of the interaction with the protein. So this is kind of weird, that the protein almost thinks it's binding to another protein because these side chains resemble side chains from amino acids. Whereas you've still got the DNA backbone, and that's really important because that makes this thing easy to measure. So that was fundamentally important, inventing a new class of reagents. If you're going to look for complicated biology, you need to look everywhere. You don't know which space these proteins are going to come from. So on the left you see a pie chart of out of the 1,129 proteins we measure, where do they come from? We don't know where the biological signals, the important ones, are coming from. So it's great that we look in all places. We don't actually know in advance what processes are really going to be driving our disease. Another of Larry's favorite sayings is we maybe know 5% of real biology. So there's 95% we don't know, even though we think we're clever. So we need to look at all of these processes. And this is the way the assay works, this thing. And I've actually looked at this, and I think it's an intelligent machine. And the reason I'm asserting that it's intelligent is because it takes a difficult problem, which is counting protein molecules, and it turns it into an easy problem, which is counting DNA molecules. And it does that by mixing together the DNA and the protein so that the reagents, the DNA reagents, find the protein that they were designed to bind to. And that enables you to wash away all the proteins that didn't find a reagent and all the reagents that didn't find a protein. So at the end of the assay, you're just left with complexes of DNA to protein. And so now you can knock those complexes apart and you can count the DNA. The sequence of each of the reagents is unique. So the sequence tells us the identity of the protein. And when we measure 1,000 of these things together, we use a chip that we get from Agilent, who's one of our major collaborators. On that chip is pre-printed the complementary sequence of each of the reagents. So they all have a known sequence. If you print the opposite sequence on the chip, they will bind, the reagents will find their complementary sequence and bind to it. So the position of a spot on the chip tells us the identity of our reagent, which tells us the identity of the protein that it was designed to bind to. And the fluorescence, the intensity of light here on the chip, tells us how many molecules bound. So I'm calling this a transducer because we've turned a protein kind of numeric counting problem into a DNA numeric counting problem. We've made it much easier because people have invested billions of dollars in making DNA easy to count, and that's what we use to measure proteins. And these are the characteristics of the assay, which are pretty unique. Any one of these characteristics you could copy with a different technology, but this combination is absolutely unique. You can find other techniques which are sensitive, other techniques that measure lots of proteins, but not both. And this has enabled us, as I'm going to go on to show you, to run the largest discovery study in cardiovascular health using protein measurements that's ever been performed. So if we want to slay the Gorgon in cardiovascular disease, what are the attributes of success? And I've listed them here. We'd like to be able to predict the future. We'd like to be able to say who's going to have an event. We'd like to say what the event's going to be. We'd like it to respond to change. If someone loses 100 pounds, goes on an exercise program, actually starts taking their blood pressure meds and the diabetic meds, we'd like the score to change. And you can see the other important features. The point here is I'm not requiring this list of features from one biomarker. I can build this list from different groups of proteins. So this is the design of the program, which I'm going to focus the results on. So as I said, it was the largest ever study performed. So we made over 3 million protein measurements. And the assay team, many of whom were in the audience, did this in about 5 weeks. It would have taken about 5 years to do this on mass spec. So, to paraphrase Winston Churchill, never before in the field of human proteomics have so much been measured by so few. So we did 2 studies. We had a discovery study, which Peter Gans, our next speaker, was responsible for. This was a beautiful study. They collected blood from nearly 1,000 people, and they followed them up now for 10 years. But when they collected the blood, they did it in absolutely ideal conditions. They processed it within 30 minutes. They had the subjects fasted, seen at the same time of day. So as a way of uncovering the biological truth, it doesn't get any better than that. The validation study wasn't quite so perfect. It was a similar size. We chose these subjects from another 1,000 subjects from a big cohort study in Norway. But they weren't fasted. They weren't collected at the same time of day, and the blood sat out on the bench for up to 24 hours. Now, at the time, that was a bit annoying, but it actually turns out to be really helpful. Because if you're trying to make a product that will be useful in clinical practice, you don't want to actually have to do any special collection conditions. So what we're doing here is saying, if we discover the true scope of the biology, the true horror of the Gorgon, how much of that horror can we actually translate into practical measurements that we can make easily in real life? And I'm not going to go through the detail of this chart, but the key features here are we measured nearly 2,000 samples. And then there's a left-hand side, which is what we did in discovery. We separated the 2 halves. We built our model on the discovery set without looking at the results on the validation set. So when we built our model, and I'm going to show you what the model looks like, we built it entirely mathematically. There was no favoritism going on. We didn't look at the literature and say, what do people like the best? We just used multidimensional mathematics to pull out the best combination of proteins and clinical data. And then we made that model, and we tested it on the validation set. Now, as I said, some of the proteins actually don't like the collection conditions in the Norwegian cohort in the validation set, and so some of them didn't do so well. But we haven't cheated. We've kept them in so far. And so how many building blocks do we get? What is the strength of the signal in the blood for cardiovascular risk? Well, this little diagram shows you that what's interesting is there were 200 proteins that were prognostic. Nearly 20% of the proteins that circulate in your blood are affected by cardiovascular disease. That was a surprisingly large number, and 120 of them have quite a big effect on hazard, the probability of having an event. And then we used there are lots of mathematical techniques you can use to choose a coherent combination. We used a technique called lasso, which chose 17 proteins. And then we ended up being a bit more frugal, selecting back from there and saying how few proteins, what's the fewest number we can measure to get the best performance? And we ended up with 9 plus age and gender. So when we look at the 17, this is a list. What you see here is a list of the proteins, and the little bars are the hazard ratio, which is saying what's the difference in the event rate, the deaths or hospitalizations, in the people with the best scores in the top 25% compared to the bottom 25%. For proteins that go up with risk, they're on the right-hand side. And for proteins that go down with risk, they're on the left-hand side. Now, there's a sports team analogy here because this mathematical technique is not just choosing the best proteins, it's choosing the best combination. So if there are two really good ones, but they're completely correlated with one another, only one will make it into this list. But I don't like the sports team analogy because it's too nice. These proteins are associated with death and destruction. This is not a sports team. It's a group of terrorists. You've got the bomb maker, the suicide vest sewer, the getaway driver, the financier. Each of these does a different job, and that's the point. This is not a literature favorite. This is a coherent combination. This is a terrorist team, and we're like the National Security Agency, trying to find metadata and data that actually says who is involved in this process and when their next strike is going to be and what it's going to be. That's our job. But in contrast to the National Security Agency and terrorism, cardiovascular disease kills more people than all the terrorist acts in history. So what are we trying to do with this score? I've shown you the individual hazards. What you're trying to do is take a population of people that today, physicians say they're the same. The population we looked at have all had a previous cardiovascular event. So today they're all labeled as high risk. But actually, most of those people don't have another event. So you're taking a population like this, and you're saying the prevalence here is 2 out of 9, or roughly 20%. But you're actually saying everyone's the same. Actually, they're not the same. Some people are going to die, and some people are going to be fine. So the purpose of this risk model is to split these into a group which have very high risks and a group which are going to be fine. And that's what I'm going to show you next. If you followed the survival of this group, it would be flat. They would all survive. Whereas if you followed the survival of this group, you'd get someone would die and another person would die. And that's the nature of the next graphs. These are survival curves. And the purpose of the risk score, remember, is to split the population into good and bad groups. We've actually used 4 layers here. On the left, you see the Framingham score, a version of it which is specially designed to apply to this population. And you can see that it's not very good at splitting the population. The difference in hazard between the good and the bad groups is about 2 to 3-fold. In our discovery set, the really well-collected samples, we can split the good and bad groups by 10-fold, and in the validation set by 6-fold. So we do a lot better job. But we're still treating this as populations. Let's look at individual people, because this is about personalization. So here we're not looking at bars or graphs or lists. Each spot on this graph is or was a real person, because unfortunately some of the purple spots have died. The green spots are people who had no more events, and the purple spots are people who had events. What I'm pulling out here is, if we look at the protein risk and we compare it with the Framingham score, how do we measure up? Well, the point here is if you look at the bottom right-hand corner where Framingham does a good job, but we missed them, it's hardly any people. But if you look at the top left-hand corner, where the protein score is really finding catastrophic risks above 50%, we're finding nearly 20% of the population lives there. And the average time to event in these people is 2.5 years. And if you're up here in the top 10%, the average time to event or death is 10 months. And when I showed this at the NIH, the 2 cardiologists came up to me afterwards and they said, we have got to find a way of helping those people. Today, these people are treated all the same. Surely there is something we can do that will be different for the top and the bottom. And so that's the Gorgon. The beauty is, of course, you can actually find normal biology in these people, and you can look at the other end of the population. And you can say, instead of labeling all of these people high risk, there are some people who actually are have a lower risk than the average person. Who are they? And they're down here. The proteins would be at the bottom here. And you can see we can find, again, nearly 20% of the population who never had another event. But today they're treated, a person down here is treated the same as a person up there. Whereas Framingham finds nobody. The next thing we're working on is to predict the type of event. We've talked about making a universal predictor. We've looked for proteins which didn't care what the event would be. We're just trying to find people who are at high or low risk. But there are proteins which do care which event. And that's what this little diagram's about. If you train a model to find specific types of events, what do you find? And what's interesting here, you find unique proteins. Each of these numbers is how many proteins on the list. The unique proteins are the ones that were not shared with any of the other types of events. And what's interesting is there are more unique proteins than there are shared proteins. And so what this tells us is that maybe we can build our product, maybe we can build this thing up to tell you who's going to have an event, when it's going to be, and what it's going to be. The next thing we want to do is to say, well, as I mentioned earlier, if someone loses 100 pounds and goes on an exercise program and takes their meds, we'd really like those proteins to change. And if someone gets years closer to their event, we'd like their score to go up so that they get a warning. This isn't lifetime risk. This is about what's happening right now. And what I'm showing you here is some early data that shows responsiveness to change. Because in our discovery study, about 500 people had another sample 5 years later. And some of those people had an event after that sample. So now we have two samples, minus 5 years, and then 5 years closer to their event. And we can say, how many proteins are there that as you got 5 years closer to your event, how many proteins changed? And these are little bars of responsiveness to change. There are a lot of proteins that are responsiveness to change to proximity to event. Some of the proteins that we included in our original model do that, but not all of them. So now we have the choice, before we finalize our model, to build in some more responsive proteins. And that's one of the things we're working on this year. The next thing I want to talk about is, well, underneath the hood here, when we're looking at the nature of the serpents on the Gorgon's head, can we learn something? And what you're looking at here is, remember I said there were 200 proteins that moved in relation to prognosis. This is those 200 proteins. This is a heat map of correlation. So the proteins in blobs are ones which move together. And you can see that they're actually they're not all in one blob. There are a set of little different blobs, and each of them represents some different piece of biology. The proteins in one blob are not correlated with the proteins in the other blob. And the biggest blob so far is kidney function, which isn't really surprising, because as the kidney changes, it filters lots of proteins. So we're only just starting to learn what this means, and this will be the basis for future research. But the promise of this is, if you find for an individual person that their risk is driven by this and this, you might be able to choose a therapy which was specific to those processes and which didn't treat something irrelevant to that person. So these are the serpents that we can see so far on the Gorgon's head, and I've talked about each one of those. So what I've talked to you so far then is what a great sensor that Larry and the team have built. But to make this actually useful in medical practice, some things need to happen. And these are the things I'm going to talk about, why we need an IT system, and unfortunately, how some of the very human obstacles are in the way of actually changing health and changing medicine. The first obstacle, remember this problem I showed you here, this objective of a test, to split a population. Problem number one is that actually if you give doctors the information, if you say, I'm going to give you the background prevalence, I'm going to tell you the performance of the test, and then you ask them to say, if the test was positive, how many people are actually going to have an event? What would it really mean? 90% of doctors get the answer wrong. And the average error is a 10-fold overestimation of risk. So that's problem number one, and that's for one test. Now imagine how you magnify that problem when doctors are checking off 5, 6, 7, 8, 9, 10 tests, or if you give them a list of 1,000 tests. So that's why you need an IT system. But, so we've learned that humans aren't very good at calculating probability, and I think the little etude before we began is going to show us that. So what do physicians do instead? Well, they use their instincts. They don't play the probability game. They play consequences. So they look at how bad it would be if they got a diagnosis wrong, or how bad the disease would be. And they like being in control. So we have to, if we're going to impose an IT system and decision support, somehow we have to help physicians overcome these issues. And the regulators aren't any better. As I've shown you, that if you give physicians those numbers, they get the calculation wrong 90% of the time. If you don't give them the numbers, the error rate must be 95%. So if you're a regulatory body and part of your mission is public health, wouldn't you think that if you know the existing system has an error rate of 95%, you'd think, I'm going to incentivize diagnostics manufacturers to actually predict risk so that the doctors don't have to do it. Somehow I'm going to help them. Is that what happened? Actually, no, the opposite happened. A special extra bit of regulation was imposed for combinations of diagnostic tests that predict risk for very human reasons, because the FDA doesn't get blamed when a physician gets a calculation wrong, but they do get blamed if a mathematical device that combines measurements to predict risk gets it wrong. So it's about human issues. So we do think that the future of healthcare looks like this, that you will use proteins and other information, that you'll integrate them together with an IT system. And then somehow you've got to help the patient and the doctor understand what's happening. And then you've got to do things which change the diagnosis or management of wellness, or the resources you apply to different pieces of the population. And how we're going to intend to do this is that we think that SOMAScan itself can become a product. And we'll start with some functionalities, many of which I've told you about here. But that's not the end, because there's a lot of real estate on the chip that today we won't be using. So what we'd like to do is to launch a hybrid product which has functionalities that are useful today and space for future learning, for future applications, which are illustrated here. So think of it like the iPhone. You get it with maps and email and other stuff, and then you get other apps from other places. So we'll develop some apps, but the world out there, we're not clever enough to develop all of the apps that are going to be useful. The world out there is going to develop apps and put them on SOMAScan. So that's the dream, that you'll work from where we're ready to start experiments in health systems with over here, but our dream is that we will fill this space up. And the combination of attributes we'd like our launch product to have is this stuff. I showed you the cardiovascular risk. We can also measure the other things. But the key question here is, if you give this information to the care team and to the patient, will they actually be able to do anything useful about it? If you tell someone that actually you are nearly certain you're going to have an event and it's going to be 10 months from now, is that going to change their behavior, or are they just going to pick the remote control back up and switch channels? And the way we're going to find that out is like this. We're talking to several different health systems about this kind of study. We'll screen a bunch of people. We'll find that cohort of people who are at very high risk, and then we'll randomize half of them to receive intensive treatment. What is that? Well, the health system will decide. Is it a nutritionist? Is it gym membership? Is it free food of a particular type? Is it a practice nurse that's calling them up every week to make sure they're taking their medications? I don't know. We'll work with the health systems to work out what that is. So the key is, if you give this information and you compare these people to the rest, do lower costs and improve outcomes. So back to the Gorgon. I think we found some interesting serpents here that are part of the inherent monster. Unfortunately, we're adding some of our own serpents to the Gorgon's head. Some of these serpents are us. So I'm going to round out here. So measuring many proteins is actually very helpful. It enables you to build a coherent model of beautiful biology or horrible disease. And these things can be powerful. but they're not enough on their own. A list of 1,000 or 5,000 measurements is not going to change health. We've got to couple that with an information system. We've got to work with physicians and care teams, and we've got to work with the regulators. So I'm going to say thank you, first of all, to the people who are far-sighted enough to collect samples, thousands of samples, so that we don't have to wait 10 years to get 10 years of follow-up on these people. So the collaborators at UCSF and at Hunt. And the SomaLogic team, as I said, this was the biggest proteomics study ever done so far. And then bioinformatics, we don't find these patterns. They don't just fall out. The team, especially David Sterling, has to extract these things. These are not simple things. There is not a well-trodden path here. And then the medical group, I've told this story as if it was easy and obvious, but actually we've struggled to decide how to do this and what to do. So as you'll see from this statue, our intention, this is a statue of Larry Gold, which those of you who have seen him with his clothes off will recognize that we had to rescale some bits of his anatomy so as to avoid public offense. But this is the aspiration, that one day we'll slay the Gorgon of disease. Thank you.
Thank you, Steve, for a great talk. We have a little over 5 minutes for questions. So where shall we begin? Let's begin right here.
So you didn't mention the usual confounding variables, smoking, gender, ethnicity. What did the population actually look like in these two groups? And did you take that into account, or is that for the future?
We did. So the population was pretty typical. The UCSF population was pretty typical of UCSF. It was run by the VA, so you can imagine the usual population that they would see. So the question is, if you make the clinical variables compete with the proteins, how many of them successfully compete and how many of them don't? And what we found so far is that most of them just are nowhere near as good as the proteins. But we did include age and gender in our model. But those are the only commonly measured clinical variables that improve the performance of the model above proteins alone. So we have been fair and looked at whether other things would change the performance fundamentally, and they don't.
Other questions?
A half comment and then a question. The answer is not so obvious, but I'll like to provoke a discussion anyway. I pick up from your comment on multivariate analysis, or multivariate vector view, if you will. And there's an insight I had from some 20 years ago in talking to a guy called Gert Grenner, who's now the Chief Technology Officer at Roche. I accidentally used the word unique biomarker in a conversation. And he said, quatch, stop, nothing. And he goes, nature is opportunistic. Nature is not stupid to pick one marker for anything. So inherently in the complexity, that non-reduction, if I could call it, it's multivariate. What can we do to educate the regulators? What mechanisms could be put in place? And it's a long marathon, it's not a sprint. You have some thoughts on that?
Well, so I absolutely agree that it is multivariate. The model we've made isn't the only model that would perform like that. There are actually thousands of combinations of the same or different proteins that would perform just as well. But what we found is it's hard to beat the performance of the model we've chosen. And in practice, when you're going for a product, there's a kind of competition between the cost of measuring things and the number of things that you want to measure. If SOMAScan becomes the product, then of course we can use as many proteins as we like. But I think the point here is that there aren't, in cardiovascular disease so far, we found these 200 proteins that appear to relate to prognosis. But there aren't 200 independent pieces of biology. Many of those proteins are correlated with one another, and there are fewer biological subspaces than that. And you only need to have a couple of proteins from each subspace. You don't need to have all of them. Yeah.
Thank you, Steve. Yes.
Thank you, Doctor. It's been estimated some 5 million baby boomers have hepatitis C. I've since cleared my hepatitis C, but for the baby boomers that are trying to get over their disease condition, as far as following the fibrosis, I was noting one of your PowerPoint maps there, and I was just kind of curious. When I was going through treatment, it was liver biopsy, which was kind of painful, and then later on they developed a FiberSure test, a French doctor. But there was a lot of debate about how accurate that was, and I was just kind of curious. I know you're more of a statistician, but in your model, how would you actually track the fibrosis in those infected or liver damaged patients? Thank you.
Yes, so you've pointed out the issue with liver damage is that the truth standard today involves a biopsy. And if you could find a signature in the blood that mimicked the biopsy results, then you wouldn't have to do that so often. And that is the program that we are in the middle of today. We have blood samples from people who've had biopsies, and we've found a signature of the amount of fat in the liver and the amount of fibrosis. We haven't done it in hepatitis C yet. We've just done it in the fibrosis associated with obesity. But the program will ultimately include other causes of fibrosis.
All right, let's move to a part of the room we haven't heard from yet. How about somebody in the back? Way in the back in the green. Oh, I'm going to make him run up the stairs.
I'll change the focus of question, actually. It's more on the entrepreneurship side. For example, myself, we're trying to put decision-making the same way you guys are trying to put decision-making onto healthcare, as well as on the large decisions on how physicians should use our data. The challenge is incredible on both regulators, FDA side, as well as entrepreneurship, making money on the bottom line. How do you envision on including FDA and the whole decision-making into the healthcare, and what are the main challenges that we're facing today, and how can we overcome together or alone?
Mm-hmm. So I think that with the FDA, I've collaborated with the FDA in the past, and I think the key there is to build trust by actually collaborating with them. When we get these studies going in health systems, we would like to bring the FDA in some capacity to actually collaborate on the projects so that they don't feel that they're being blindsided or ignored. And they've created an innovation pathway. Maybe they would put this into an innovation pathway. But clearly, the regulation today that regulates one marker at a time, when we're actually going to be measuring thousands, isn't going to fit. So we do need to find a new way of working with them. And then the business model is, today, a doctor checks a box on an order form, and that goes out, and the diagnostics company makes 25 cents when they run that test. How will that work when you're applying this kind of system as a surveillance health wellness management program? And again, talking to these integrated health systems, the signals from them today seem to be that if you can show them that they save money and improve outcomes, then they'll just treat you like an IT supplier, like a systems supplier, as they have contracts with many systems suppliers today. They buy electronic medical records from Epic. They pay Epic an annual fee for the provision of EMRs. So there might be another kind of business model that would be more appropriate to this approach than the traditional diagnostics one.
One more question. Let's go to this side of the room. Yes, in the white shirt.
Yes. What's the influence of 1% renal function on the markers, on the actual numerical value of the markers?
So the question is, what's the influence of a 1% change in renal function on our markers? And we have done a big study in kidney function where, in collaboration with the University of Lund, where they had measured true renal function by injecting a dye. So they knew what the glomerular filtration rate is. And we've asked, how many proteins are affected by big changes in kidney function? And the answer is a lot. Probably hundreds of proteins are affected by kidney function. And so what we're doing about that is to actually create a little panel of proteins which is better than any of the existing protein measurements, particularly up in the normal range, to actually create a better measure of actual renal function. And we're going to incorporate that, or we're going to see whether incorporating that into our model actually improves the performance or not. And some of the proteins that are already in the model will be surrogates for changes in renal function. So in a way, because kidney function is so important, and the mathematical model chooses from all the array of proteins it has to work with, some of those proteins will be in there because they capture that effect. But the question is, if you actually use an even better model, will you be able to improve the performance more? And we'll see.
Thank you, Steve.